In this video, we're going to talk about the steps of phagocytosis. The first step in phagocytosis is chemotaxis. Chemotaxis is the chemical attraction of the phagocytic cell, here that I have in white, to the site of infection. So we have damaged tissue. Our cells, they release what are called cytokines. So these phagocytic cells are going to be naturally drawn from an area of low concentration of the cytokine to an area of high concentration. So it's a chemical sensing, more or less. So the phagocytes are going to move towards the site of infection. Second step would be recognition and attachment. The recognition portion comes usually by the fact that the bacteria or the other antigens would be coated with antibody. Antibodies interact in a specific manner to antigens. So the antibodies Fc stem attracts or flags this bacteria for phagocytosis. The second portion, attachment, comes into play where the actual phagocytic cell, they have Fc receptors. So these Fc receptors attach to the Fc stem of the antibody. Step 3 would be ingestion. So after this bacteria attaches to, or the phagocyte attaches to the bacteria, this phagocyte is going to form a vesicle, and that vesicle is called the phagosome. And that phagosome will include that bacteria. So I'm going to draw a new phagocyte here. So phagocytes are irregular shaped cells, which is why I'm going to make them a little weird shaped. So ingestion, and we get the formation of the phagosome. Step 4 would be digestion. And what has to happen with digestion is there's other vesicles inside the cell called lysosomes. So these two vesicles, the lysosome and the phagosome, have to fuse together. The lysosome contains enzymes and digestive chemicals that will actually work on the bacteria. The phagosome at this point does not have any of these enzymes or chemicals to act on the bacteria. So we need to have these two fused together. So these two vesicles will fuse together and form what's called the phagolysosome. And now we have the enzymes and the chemicals with direct access to the bacteria. So these enzymes and chemicals will digest this. Step 5 would be release. Release of the digested bacteria. So all the bacterial particles that are broken down and whatnot will be removed through a process of exocytosis.